Hello everyone, welcome back once again to my educational channel on biology. Teacher Janet here. In this video, we'll discuss the MELAKA 2025 Paper 2 questions. They are very good questions. And we'll discuss the structured questions first of all, question 1 to question 8. Alright, so this paper 2 is a challenging paper where students may find it a challenge to obtain a good grade or an A+ grade for this paper. So practice makes perfect. And also, genius is 1% inspiration, okay, 1% the intelligence or brilliance of the mind, okay, and 99% perspiration. Perspiration means hard work, where you practice, you study, and have consistent study time and revision. All right, so 99% perspiration. This saying is attributed to Thomas Edison, who invented the light bulb. Okay, so we need to set our hearts to persevere and prepare ourselves for the exam by dedicating time to revision and memorization. Right now, I'll be continuing also with my videos on the 10 winning strategies. Okay, but in the meantime, let's try some of these good questions that can prepare you for the SPM biology paper. So let's start with the first question. Diagram 1.1 shows the villus structure in human digestive organs. That is the ileum. All right, the ileum contains the villus. So this first question is based on Form 4 Chapter 9 on digestion. Now name cell R. So you can see the layer of cell R here. Okay, it's one cell thick layer. So R is on the surface of the villus, so it must be the epithelial cell. All right. Then next, take two characteristics found in the villus or villi for efficient nutrient absorption. So characteristics are often tested. You're asked to just state, all right, and it must be a characteristic that helps in efficient nutrient absorption from the lumen intestine into the villus. All right. So by looking at this picture, you should be able to guide you a bit, all right, on what to remember what the characteristics are. Firstly, the layer of epithelial cells. It is epithelial layer of the villus is very thin, only one cell thick. That's the first characteristic. Now, do you need to give the reason why? So here is just to state the characteristics. So this is enough for one point. But if you're not sure, just write in. Okay, write in here the reason why. So the epithelial layer of the villus is thin, only one cell thick, to allow rapid diffusion of digested food substances into the villus. Okay, to allow rapid diffusion or quick diffusion of digested food substances into the villus. Now, secondly, goblet cells secrete mucus. So goblet cells are found in the layer of epithelial cells here, and they secrete a sticky, smooth substance called mucus. So what's the reason? To lubricate the movement of the food in the small intestine, right? To lubricate the movement of food in the small intestine. Now next, another point that you can write in is villus contains a network of blood capillaries as shown here. Now reason is for what? To transport glucose and amino acids, right? To absorb and transport glucose and amino acids. Okay, and then it also has one more structure inside here, which is called the lacteal, L-A-C-T-E-L, T-E-A-L, L-A-C-T-E-A-L. So lacteals help in the absorption and transport of the fatty acids and glycerol. Okay, so any two characteristics will give you the 2 marks. Now, if they ask explain, if they state explain, then you have to give the explanation. What's the function of each characteristic? They say explain a characteristic found in the villus for efficient nutrient absorption. Okay, so explain is probably 2 marks. Now, diagram 1.2 shows the organ that is involved in assimilation of digested food. Okay, so this is the liver, right? This is the organ, and here you have glucose converted to glycogen, and also in the liver cells we have this body cell or liver cell. Okay, now based on diagram 1.2, explain the assimilation process that occurs in the organ, that means in the liver. So as you can see here, you can be tested on the names of the cells or tissues, the characteristics, and then also the processes. All right, so but in this case, based on this diagram, is only related to glucose, not amino acids, just glucose. 3 marks. So 3 marks is quite detailed, all right? Now, so first mention what happens in the liver cells, all right? How is the glucose used? So in the liver, the glucose is oxidized in cellular respiration to produce energy. Okay, so this is F. This is compulsory point, so F. And then, secondly, what happens to the glucose? This way, it is converted to glycogen when the blood glucose level is higher than normal. Okay, and the blood glucose level is higher than normal, for example, after a meal, right? So the glucose, excess glucose, now you must mention excess glucose is converted to glycogen. If you just write glucose is converted to glycogen, you'll get it wrong. So they like this word excess, make sure you write excess blood glucose is converted to glycogen when the blood glucose level is higher than normal, okay? And then the glycogen is stored in the liver. So this is another point, okay? And then separate part is when blood glucose levels decrease, or when the body needs energy, when blood glucose levels decrease below normal, then the glycogen can be converted back to glucose, right? So here, F is the first point that's compulsory, and then F with P1 and P2, or F with P3 and P4, okay? Now, if you're not sure how far to write, how many points, just write a bit extra to make sure that you get the complete marks, all right? Question 2 is on chemical composition of a cell, chapter 4 in Form 4 syllabus. Diagram 2.1 shows the equation for the formation of Q in sugarcane. So take note, it's in sugarcane, all right? So what is Q? Now, glucose plus P through the process of condensation produces Q plus water. So this is a condensation process whereby two monosaccharides combine together to form a disaccharide. So Q must be sucrose. Okay, the disaccharide in sugarcane is sucrose. It's the white sugar that we use to sweeten our drinks. If Q is sucrose, what is P? P is fructose. Okay, now to remember this, remember the reverse reaction, which is hydrolysis. Sucrose is broken down by water to form glucose and fructose. Think of the word sugar-free. As for sucrose plus water, arrow becomes G, sugar-free. The G is for glucose, and then free, F. The F is for fructose. Okay, think of sugar-free. So, but this is the reverse process. So you have to reverse that process there. Now, a student carries out Benedict's test on sample Q, which is sucrose. Explain the experimental observation. So when we boil the sucrose with the Benedict solution, we heat it up, not boil it. When you heat it up, does it give any positive observation? No. No brick red precipitate is formed, first of all. Okay, because sample Q, don't write sucrose. Here they use sample Q, so you use sample Q also, is a non-reducing sugar. The sucrose is a non-reducing sugar. Okay, out of the 6 sugars, like glucose, fructose, galactose, maltose, and lactose, only sucrose is the non-reducing sugar. What does it mean? It means that it does not react with the Benedict solution or the blue copper sulfate in Benedict solution, and it does not reduce that blue copper sulfate solution to form the brick red precipitate of copper 1 oxide, from copper 2 to copper 1. It cannot reduce the copper 2 to copper 1. Okay, that's why it's called non-reducing sugar, right? So any of the 3 points here, it is 3 marks, all right? Now, diagram 2.2 shows one nucleotide unit. So the nucleotide consists of a phosphate group, a pentose sugar, and a nitrogenous base. So if you have a column of nucleotides here that are joined together, it forms the polynucleotide strand. Okay, that's found in DNA and RNA, the 2 nucleic acids. So here in the space provided, complete the other side of the nucleotide structure, especially for DNA molecule. It has 2 polynucleotide strands, one on the left here and the other one on the right. So how is the position of the nucleotide on this other side? Okay, have you drawn? Let's see whether the structure is correct. So here in this case, imagine that this nucleotide is rotated into this side, right? So the pointed end of the sucrose, of the pentose sugar, the pointed end of the pentose sugar, should be in opposite directions. That's the first thing. Okay. And then, okay, let's read from here. Garisan bersambung. All these lines should be continuous. Okay, no broken lines. Ikatan kumpulan fosfat dan base bernitrogen selang satu karbon. So this bond here, there's in between this bond and this bond, there is one carbon atom here. All right. So you cannot draw the phosphate group combined with this part here. With the bond here. Right. Next, bentuk base bernitrogen mesti sepadan dengan pasangannya. So that means the bases are complementary and the shapes are also complementary. They match each other. If this is rounded, then this side also must be rounded. Okay, and it must match together. All right. So this is called the antiparallel arrangement, where this strand of polynucleotides, when they're joined together, they are parallel to the other side, but it's antiparallel means that this one, the pentose sugar faces upwards, the pointed end faces upwards, this one is downwards. Okay, so if you draw the pentose sugar with it on this side with the point here similar to the point here, okay, that's wrong already. Okay, now next, state one difference between the nitrogenous bases in DNA and RNA. So circle this word, nitrogenous base. What's the difference? Now in DNA, the nitrogenous base is thymine. One of the nitrogenous bases is thymine. But in RNA, there's no thymine. The thymine is replaced with what? Uracil. Okay, let's look at the answer. So DNA contains the nitrogenous base called thymine, T, but RNA contains the nitrogenous base uracil, U, in place of thymine or instead of thymine. All right? So 1 mark for that. So differences are also tested very often. Question 3 is on Form 4, Chapter 11 on immunity. Diagram 3.1 shows the method of producing substance M extracted from horse's blood. Now this diagram, there's a similar one in the textbook. All right. So here we have the titanous toxoid suspension. What do you mean by that? It means that it is a toxin from titanus, but already inactivated, meaning it's weakened, it's no longer activated. It means it cannot cause the toxin, cannot cause a person to fall ill from the toxin anymore. Okay, the toxin has been sort of weakened or neutralized already in a way. All right, it's inactivated. So next is injected into a horse. So what is the purpose? So that the toxin will stimulate the lymphocytes of the horse to start producing specific antibodies against it. Okay, so the horse will produce the antibodies against the toxin. Probably these are antitoxins which can neutralize the toxin from the titanus pathogen, right? So then the substance M is isolated from the horse serum and purified. Okay, so the antibodies will be purified, will be extracted in the serum. Serum is a plasma, blood plasma without the clotting factors like fibrinogen, so that the serum will, there will be no clotting in the serum. Okay, so that's substance M. So what is M? M contains antibodies in the serum, so it's called, now name substance M. So M is the anti-serum. Okay, antibody or the antitoxin in the serum. Okay, now next, this anti-serum can be injected into an individual who has already been infected with tetanus. So these are ready-made antibodies in the anti-serum that will cause the patient that is sick with the tetanus or has been infected with tetanus to be able to use these antibodies from external sources from the horse to destroy or to neutralize the toxins of tetanus, so that they will recover from the sickness from tetanus. Okay, so substance M is an anti-serum containing the antibodies that are directly injected into a patient. So the patient doesn't produce his own antibodies, right? It's from the anti-serum. So state the type of immunity acquired by the individual. So this is the passive immunity. Artificial, because it's through injection. Passive immunity, meaning that the patient himself or herself did not produce the antibodies. It was from external sources, that means from the horse, actually. Table 1 shows immunization of babies with substance N according to the prescribed period. Okay, now this is talking about another substance called N. Now, by the way, I think the answer given in the marking scheme was not correct. I think they wrote artificial active immunity, something like that. Now, even they don't, this is the right answer anyway, artificial passive immunity. So now we go to substance N, go on to substance N. So immunization of babies with substance N. When you see the word immunization, it means that the babies were injected with a vaccine, not anti-serum, not an anti-serum. So vaccine is different because it contains the weakened or dead pathogens or toxins from that are the real weakened pathogens, okay, that is injected to stimulate the formation of antibodies by that person who got the injection. Okay, in this case, it's the babies. So based on table 1, explain the function of N in providing protection and prevention against tetanus infection. Now, so this type of injection for tetanus is different from the first type here. Okay, remember that means there are two types of tetanus injections, two types of immunity. So for B, actually, it is for the baby to acquire artificial active immunity compared to this first case where the sick patient is injected directly with the antibodies from the host. Okay, in this case, the babies are stimulated so that their lymphocytes are stimulated to produce their own antibodies against tetanus. Okay, so read the question carefully. Function of N. So identify N. What is N? N is a suspension of weakened or dead pathogens. It is the vaccine, right? Then in providing protection and prevention against tetanus infection based on the table. So you have to talk about the dose 1, dose 2, dose 3, and then the booster dose. Okay, now let's look at the answer. First, identify substance N is the vaccine, and then give the definition: a suspension of dead or weakened pathogens or toxins, okay, that are inactivated. So next, so it's something like this tetanus toxoid suspension, but it's injected not into the horse, it's injected directly into the patient's body, into the, not patient, into the baby's body, which these are healthy babies, okay? So injections of dose 1, 2, and 3 here, any one of these, stimulates the lymphocytes to produce antibodies against the pathogens of diphtheria, tetanus, and pertussis, according to the shadow. Okay, so the babies will directly produce antibodies, and then state the function of the booster dose. So booster dose injection increases the antibody levels above the level of immunity. So the level of immunity for the antibody must be reached so that the baby becomes immune to the disease. So lower levels of antibodies are not may not help to make the baby get the protection. So they must increase the level of antibodies above the level of immunity by giving a booster dose after 18 months from birth. Okay, so then if the person or baby is infected by actual pathogens, now the lymphocytes can produce enough antibodies already. Okay, because the level of antibodies is above the level of immunity, so it's a very high level of antibodies. So enough antibodies are produced to actually destroy the pathogens before the patient can get sick. Okay, so any 3 points here to get your 3 marks. Okay, that means substance N can provide the protection and prevention against tetanus infection. So still on immunity, diagram 3.2 shows the action of virus S that attacks the blood cell T and causes AIDS. Now this is a hot question because you know this is a new diagram that's given to you, right? So what is virus S that attacks blood cell T and causes AIDS? So virus S must be the HIV virus. Okay, I can see here that it has landed on the cell and then it injects its viral DNA into the cell. Then next, reverse transcription copies the RNA of the virus to produce proviral DNA. So the DNA is produced from the RNA and it is proviral. That means it's actually the viral DNA. Okay, then proviral DNA integrates with the host DNA. So this is like an invasion of the aliens, right? The DNA of the host has been infected with the DNA from the virus. Okay, So in that way, the virus will take over the function of the cells and produce its own RNA and protein code. Then when the cell is full of these small viruses or young viruses, it will burst and die. So in this way, the lymphocyte, this is a lymphocyte, right? Because HIV virus attacks the lymphocytes. So the lymphocytes will be destroyed and the level of lymphocytes will be low, correct? So that will weaken the immune system. Now let's look at the question. Based on diagram 3.2, explain the effects of infection of virus S on a person's cell. So I like to explain this word. This is a common phrase. Explain the effects of A on B. In this case, explain the effects of infection of S on person's cell. So to phrase this question, we have to know the three aspects in this question. Firstly, there is the event. The event is infection of virus S, which is a HIV virus. Next, we have a few steps, something occurring. And then finally, the person's health is affected. In this case, the person may die from other diseases, right? So you have to explain what happens after infection by virus S based on this cycle. Okay? And then until the person's health deteriorates. So firstly, I'll suggest that you write what happens to the person's health first. State the final result. And then after that, explain. So I will go for P4 first, or P3 or P4. The patient, HIV patient, will die easily will be easily infected by other pathogens and they may die from other infections such as pneumonia. 2 marks. Okay? And then you go back to the beginning and say this is because the virus S, which is the HIV virus, attacks, multiplies, and destroys the blood cell T, which is the lymphocyte. Give the reason why. And the virus S therefore weakens the immune system. Okay, because they kill the lymphocytes. Right? So the 4 points are here, but here they start from beginning. Virus S, as based on this picture, is the HIV virus which attacks and multiplies and destroys blood cell T, which is lymphocyte. Thus, virus S weakens the immune system once the number of lymphocytes are reduced. Then HIV patient is easily infected by other pathogens and they will die from other infections such as pneumonia. Next question is on Form 5, Chapter 8 on biodiversity. Table 2 shows the taxonomy hierarchy of the Malayan tiger. Okay, so hierarchy is like different levels. Okay, different levels of the classification system, and the taxon means what actually is the different levels that the Malayan tiger belongs to. What are the names, okay, of those different levels? For example, the phylum for tiger is Chordata, meaning organisms with backbone. The class is Mammalia. Order is carnivore. Family is Felidae. Now, next, what is the level of in the hierarchy here? In the hierarchy of classification, this level is called the genus. Then finally, species. And then, what is the kingdom to which the Malayan tiger belongs? Yes, Animalia. Okay, so X is genus and Y is kingdom Animalia. Okay, the genus is Panthera. Next, state the scientific name of the Malaysian tiger. So of course it consists of the genus name and the species name, Panthera tigris, but it must be written according to the rules like this. Panthera, okay, underline it, and then tigris, underline. And the first letter of the genus name, Panthera, the P must be a capital P. Okay, now if printed, it should be in italics without underline, Panthera tigris, right? But if it is written by you, that is Panthera, underline the word Panthera, and separately underline the word tigris. And then tigris, Panthera starts with a big capital letter P, but the tigris with a small letter T. Next question. Based on your answer in 1A2, explain the binomial nomenclature system rules. Okay, so there are some rules for this system of name. Now, any 2 or 3 of these are according to the marks here. Scientific, any 2 points. Scientific name consists of 2 words. The first word is the name of the genus, like Panthera, and the second word is the name of the species, which is Tigris. Next, the first letter of the genus is capitalized. You can see the big P here for Panthera. And the name of the species begins with a lowercase letter, Tigris with a lowercase letter. Scientific names must be printed in italics as shown here, okay, without underlining it. And if handwritten, the two names must be underlined separate. So underline then separate. Okay. Question B, diagram 4 shows names of different organisms such as the bird, bat, whale, cat, horse, and humans. Based on diagram 4, what is the structure used in the phylogenetic classification? So phylogeny is a new topic in the biology syllabus. All right. So this is taken from the textbook. Phylogeny means the evolutionary history of a species or a group of organisms that are genetically linked. So evolution means that there are changes in the species as through time, and then it's showing the changes, explaining the changes in the species. All right. So that the structure of the organism may change over time. All right. And also the characteristics may change. So evolution, we have to understand what is the theory of evolution. So evolution can be defined as changes in the characteristics of a species over several generations. And this relies on the process of natural selection where the fittest or the most adaptable will survive to the changes in the environment, and the weakest or least adaptable will not survive, okay, will die. So, for example, Charles Darwin studied a type of bird called the finches in different habitats, different environments. He found that they have different big adaptations according to their different food sources in their different habitats. So although they are of the same species, but they have adapted to the habitat and to their food source by having different big structures. All right. So that is the theory of evolution. Now, I do believe that this can occur among organisms of the same species over a long period of time, depending on their environment. All right. However, in phylogeny, it may go beyond just one species. Okay, it's the evolutionary history of a species or a group of organisms, such as, let's say, the different organisms here, the different species of organisms like bird, bat, whale, and cat. So they are saying that these have some similarities among each other, all right, due to the evolution process, and that they may share a common ancestor, all right. So that is the question mark there. Some people, for example, do not believe that all species of organisms share a common ancestor, even though they have similarities in their structures, right? For example, now let's look at the question. Based on diagram 4, what is the structure used in the phylogenetic classification? So it must be a structure that is similar among all of it. For example, the limbs. So this is called the homologous structures that are compared among the different species. Okay? So they claim that because of the similarities, they may all come from one single ancestor. So again, how this is actually a hypothesis only, all right, as mentioned by the textbook. It's not proven to be 100% true. It depends on what your own opinion is on this, all right. It could be that it is from the same designer, right, that has the same common idea of how to create all these structures, all right. So it's up to you what you think is the correct thing here, all right. In this case, it still remains a hypothesis. Now, state the purpose of phylogenetic classification in classifying organisms. So the purpose is to present a hypothesis about the evolutionary history of a group of organisms that may be genetically linked or are genetically linked, okay? And it presents a hypothesis, all right, that has yet to be proven. So do men or different organisms come from the same ancestor, like a single cell organism or whatever? We don't know which ancestor is, right? So it's up to you to form your own opinion on this. Question 5 on chapter 1 is on chapter 1 of the Form 5 syllabus, and is related to plant tissues. Okay, so diagram 5.1 shows a longitudinal section of the tip of an eudicoट plant root. Now, name zone P and zone R. So zone P is here, then Q, then R. Okay, as we go further down. So we can see in zone P there are root hair cells. That means zone P is the zone of what? Cell differentiation. Okay, because the epidermal cell will differentiate from root hairs in this zone. So, whereas zone R is at the tip of the root, just above the part here called the root cap. This is called the root cap that covers the tip of zone R. So zone R is the zone of cell division. All right, the first zone where cell division by mitosis occurs. Now next, diagram 5.2 shows cell development in zone Q. Okay, this zone, which is actually the zone of cell elongation. So can you draw the diagram here? Now I've used this question to also test you under the winning strategies series. All right. So if you have done that, you should be able to draw the diagram here very easily. Now these are the few stages in cell development in the zone Q. Okay, from the small cell, then what happens? The small vacuoles will fuse together to become bigger and bigger vacuoles. At the same time, water diffuses into the cell by osmosis in order to enlarge it. Then the water is stored in the vacuole, and the vacuole will press against the cell wall, and then the cell will expand. Okay, until finally the last picture will look like this. Right? It's just like a normal plant cell. All right, before cell differentiation occurs. So there's one big central vacuole, because all the small vacuoles have combined together to form the one big vacuole in the center of the cell. Okay, so the vacuole will press on the cell wall, and it will cause the pressure will cause the cell to elongate. Right, complete diagram 5.2 by drawing the formation of a central vacuole due to diffusion of water by osmosis in the space provided. This is what you have done here. All right, draw the central vacuole. Actually, this is a very specific, really very clear, and it helps the student to answer the question very easily. So number 2, name the process in diagram 5.2. What is this process? Can we still write cell elongation? It's not mentioned, but actually, to be more specific, the process here relates to the formation of the vacuoles and then the fusion of the vacuoles, and the vacuoles fuse together or combine together to form one large vacuole. So the answer is, the process is vacuolation, which occurs during cell elongation. All right, if you're not sure, you put which occurs during cell elongation. Next question, diagram 5.3 shows a cross-section of a plant stem that undergoes different types of growth. So actually two plant stems, okay, of two plant stems. So this plant stem A, plant A, you can see here that it is the cross-section of a stem showing only primary growth. So the two types of growth are primary growth and secondary growth, right? And in plant B, you can see the formation of these secondary tissues like secondary xylem here and also secondary phylum outside, all right? And then you can see the cork cambium layer forming here, all right? So plant A shows primary growth and plant B shows secondary growth, right? So based on the diagram 5.3, differentiate the type of growth based on the criteria in table 3. So the criteria is mentioned here. Meristematic tissues involved and the effect of the growth on the plant, right? So you can fill this up first and also answer this question. Right, let's look at the answer. For plant A, which shows primary growth, the meristematic tissue involved is the apical meristem at the shoot tip and root tip that causes the growth of the plant vertically. Okay, the stem will grow upwards, grow longer, okay, and the roots will grow longer downwards, right? Whereas for plant B, the meristematic tissue involved in secondary growth is the cambium. So it's the lateral meristem as opposed to the apical meristem. So example is the vascular cambium in between the xylem and phylum. Okay, this one, this ring, and also the cork cambium, which is on the outer part of the stem. Okay, right. Next, effect of growth. Effect of this primary growth on the plant. So it causes the length of the stem to be increased. All right, and the length of the roots also to increase. Increases the length of the plant stem and roots. Now for B, plant B, secondary growth, it will cause lateral growth where the stem diameter will become bigger and bigger. Okay, so it increases the diameter or circumference of the plant stem so that the width of the stem will be bigger. Okay, now if we write width is accepted, so in the textbook they use the word diameter or circumference, which is more accurate. All right, so it increases the diameter or circumference of the plant stem. So next, plant B can live longer than plant A. Explain the advantages of the type of growth experienced by plant B that can increase its lifespan. Advantages is a common question, all right, of the type of growth. When you see advantages, you have to use the comparison mode, like more of this and more of that. So type of growth experienced by plant B. So the type of growth is secondary growth. So if you were to rephrase the question, it would be explain the advantages of secondary growth, okay, by plant B that can increase its lifespan. So what are the tissues that it produces during secondary growth that help it to increase its lifespan, right? Okay, so 2 marks, so any 2 points. Now let's look. Firstly, this type of growth provides more mechanical support and stability to the plant as it grows. In what way? Because more secondary xylem is formed, which will give more support to the plant stem, right? So that it will not topple over or it will not break in the center, okay, in the center of the stem. And stability too, because the roots will also undergo secondary growth. So this will help the plant to grow without toppling over and dying. Now, P2, it produces small secondary xylem and phloem tissues to replace the old damaged xylem and phloem tissues. So as the plant grows older, the old xylem and phloem tissues may be damaged. So secondary growth helps to produce even more xylem, called the secondary xylem, and the secondary phloem tissues to replace the old ones, so that the plant can continue to carry out the functions, okay, its functions well, such as the transport of substances, right? So P3, secondary growth produces stronger and thicker bark to protect the plants from excessive water loss, physical injuries, or pathogen infections of pathogen. So secondary growth produces a thick bark on the outer part of the stem, stronger and thicker bark to protect the plants from excessive water loss, and so that the insects or pathogens cannot easily attack the plant stem. Okay, so any 2 of these points. And take note, I've used the comparison mode here. It provides more. If you just say it provides mechanical support, not correct, because even primary growth does provide some mechanical support through the lignified tissues like the xylem tissues. Okay, you must add the word more xylem and phloem, because in primary growth there's also formation of the primary xylem, right? But this one, it produces secondary xylem, so there's more xylem, okay, and stronger and thicker bark. Comparison mode, very important as part of the technique of answering. Question 6 is on transport in humans from Form 4, Chapter 10. Diagram 6.1 shows the parts of the blood circulatory system and the lymphatic system in humans. Okay, so the blood circulatory system consists of this blood vessel, which is probably the arteriole, that branches out to form the blood capillaries. And then after that, the blood will flow into the venu, okay, and then back to the heart. Then apart from the blood circulatory system, there's the lymphatic system, which consists of these tubes, all right, with the closed ends. So this is the lymphatic capillaries. These are the lymphatic capillaries because of the closed ends, right? And so we are asked to name the fluid X and fluid Y. Fluid X is in the blood capillaries, so it must be blood, right? And fluid Y, which is in the lymphatic capillaries, it must be lymph, okay? So label tissue fluid in diagram 6.1. So this is another fluid. Apart from blood and lymph, you have tissue fluid in this diagram. Where's the tissue fluid? The tissue fluid is the fluid in between the cells, right? So you can label somewhere here, label this empty space here with not an arrow or with a line, okay? Don't draw an arrow because it's wrong. You just use label lines that are like this. So use a ruler to draw the label line. Don't just draw it freehand, okay? And then write the word tissue fluid, because they ask you to label tissue fluid. Unless they ask you to label X as tissue fluid, then you have to write X. But here it's just label tissue fluid. So follow and read the question carefully before you answer. Okay, don't try to do it too fast. Read the question a few times to make sure you know what it is, and then quickly write it down. All right. Number 3, compare and contrast fluid X and fluid Y. Now, when you see this word compare, you must have at least one similarity and one difference. Since it's 2 marks, you should have one similarity and one difference. If you write 2 differences, you only get 1 mark. Okay, you should have at least one similarity. And if you write 2 similarities, you still get 1 mark. Okay, so you must have one similarity and one difference. So what's the similarity between fluid X, that is the blood, and fluid Y, which is the lymph? Now, in this case, we can use the term both, right? The similarity is that both have the same thing, both fluid X and Y. Can we say both blood and lymph? No, you follow the question. Question refers to the fluid as fluid X and Y, not blood and lymph, right? So use the word fluid X and Y. So similarity is that both fluids contain nutrients such as glucose and amino acids, hormones, enzymes, cellular waste, respiratory gases, and leukocytes. All right. So actually, these substances have diffused into the intercellular spaces to form the tissue fluid, right, from the blood. But then after that, some of these nutrients, hormones, enzymes that are remaining, that did not diffuse into the cells, will then go into the lymphatic system. Okay, so they are found in the lymph too, although in smaller concentrations, especially for oxygen, glucose, all those have diffused into the cells. So there may be some remains of this glucose and oxygen, so it will go into the lymphatic capillaries, so their concentration may be less. Okay, the level of the oxygen and glucose is less in the lymph compared to blood. Okay, but carbon dioxide is more, okay, in the lymph than in the blood. All right, so let's look at the differences now. So there are many differences given here. Just one of it will be enough. All right. Now, firstly, we know that certain components in the blood cannot diffuse into the intercellular spaces because the items are too large in size. They cannot diffuse out through the capillary walls, the pores in the capillary walls. They cannot go through that pores. So there are 3 items or 3 components of blood that cannot come out from the blood capillary, and they remain in the blood capillary, so they won't be found in the lymph. So these are, now you can write fluid X contain plasma proteins, but fluid Y, the lymph, doesn't have. Okay, because these molecules are too large to pass through the wall of the blood capillaries. Another one is the erythrocytes or red blood cells. Fluid X contain erythrocytes, whereas fluid Y does not. And fluid X contain platelets, whereas fluid Y does not. Okay. Now, then other than that, fluid X is found in the blood vessels, the location. Okay, that you can give differences based on location. So fluid X, the blood, is found in blood vessels, and fluid Y, which is lymph, is found in the lymphatic vessels. Now, another difference is fluid X contains more oxygen, less carbon dioxide, compared to fluid Y. Fluid Y has less oxygen and more carbon dioxide. This is the waste product from the cells. Okay, the carbon dioxide. Next, fluid X has higher glucose content compared to fluid Y, because some of the glucose will diffuse into the cells. So fluid Y, which returns the tissue fluid back to the blood, it will have less glucose already. Some of the glucose has diffused into the cells. Okay, fluid X has low lipid content compared to fluid Y. Why? Because fluid Y is in the lymphatic system, and the lymphatic system transports the lipids from the lacteals in the small intestine back to the blood. So it has a lot of lipids from the small intestine, from the lacteals in the small intestine, that absorb the lipid soluble substances, okay, and the final products of lipid digestion. Now, fluid X has less lymphocytes than fluid Y. What's the reason? Because lymphocytes are produced in the lymphatic system, okay, and that's why there are more lymphocytes in the lymphatic system. Okay, and then next, the color is different. Fluid X is red in color, fluid Y is pale yellow. Right, question B, Diagram 6.2 shows a type of worm which is transmitted by mosquitoes to humans. The mosquito is the vector of disease M. Okay, so you have to find out more about this picture. First of all, a type of worm transmitted by mosquitoes to humans. So it's a parasitic worm, and the mosquito is the vector or carrier of disease M. So if you look at disease M, it shows a swollen leg. All right, so these are my extra notes. Actually, the disease is lymphatic filariasis. The common name is elephantiasis because the leg looks like an elephant's leg. All right, and then according to our notes, it's caused by the parasitic worm called Brugia species, which infects the lymphatic vessel. So it will block the flow of lymphatic fluid in the lymphatic vessels in the leg, and the infected part, such as the leg, will swell. The patient contracts the disease called lymphatic filariasis, and the worm is transmitted by mosquito bites. Okay, so mosquito transmit worm larvae while biting human, and then worm larvae reproduce in lymphatic vessels. These are the worm larvae, and then they block the flow of lymphatic fluid, so it causes the swelling, right? And this is the disease called lymphatic filariasis. So we stick to this name, all right? It's in the textbook. Sometimes it's called elephantiasis. Now, suggest ways to prevent disease M. So this is not in the textbook, all right? So it's a hot question, but using your common sense, you'll find that you can talk about the prevention of being bitten. We must prevent ourselves from, a person must prevent himself from being bitten by the mosquito, which is a carrier. Vector is a carrier of these worms that cause the disease M. All right. So your 3 marks here is just on how to prevent mosquitoes from biting the person or from reproducing, okay, in the environment. So simple, right? So understand the question, and then you know how to answer your HOTS question. Okay, let's look at the answer. So try and write your answers first, and then we'll see. Right, 3 suggestions. Suggest ways means here is 3 marks, means 3 suggestions. Okay, first suggestion: wear long sleeves or long trousers. Now, do you have to state the reason why? It's better to write in the reason, okay, if you are not sure. So wear long sleeves or long trousers to prevent being bitten by the mosquitoes, right? And then secondly, install the mosquito net on the window or door, or sleep in mosquito net, again to prevent oneself or the skin from being bitten by the mosquitoes, right? Now, thirdly, use mosquito repellent. What's the reason? To repel the mosquitoes so that they will not bite the person, right? So simple. And the 4th point, which you can also add, is destroy the mosquito breeding ground sites. For example, stagnant water in a container, all right? So by destroying the breeding ground, we prevent the larvae from growing in the stagnant water, right? And stop the reproduction of these mosquitoes, okay? So any 3 points for 3 marks. Now, taking medicine to kill the filarial worm, I think not so good, because this one, in this case, is already, there's already infection already, all right? Whereas suggest ways to prevent disease, and it means that before the person is infected with the disease, okay? So leave out number 4. Just P1, 2, 3, and 5. Any 3 of these with the reason. The next question, diagram 7 shows the process of aerobic respiration that occurs in animal cells, right? So the substrate to process P becomes pyruvate. And then after that, this occurs in the cytoplasm, and after that, the pyruvate enters the mitochondria, all right, to be broken down further. So what is process P? Now, the process P occurs in the cytoplasm, and it is glycolysis, right? And then substrate, the substrate is glucose that is broken down through glycolysis to form pyruvate. Okay, now next, write the word equation for aerobic respiration that occurs in the cell. So think of GOWEC for aerobic respiration. That's the acronym for aerobic respiration. G for glucose, O for oxygen, W for water, E for energy, and C for carbon dioxide. So easy, GOWEC. That's the word equation for aerobic respiration. Now, word equation doesn't have any symbols or chemical formulae. So don't write O2. All right, you must write the word oxygen, for example. All right, so simple. Now, let's go on to C. So a 100-meter sprinter breathes rapidly for several minutes after finishing his race. Explain why. So analyze the question first. It's 3 marks. So before you write in the answer, analyze the question. Now, usually in biology, for this kind of questions, there's an event leading to a process and then the final effect or result. So it depends on what they want to test you on, what they expect you to answer. All right, could be something about, mention something about what occurs during the event or the process, or what is the final effect. All right, now what is the event here? The event is that the sprinter ran the 100 meters sprint, and after that he breathes in rapidly for several minutes after finishing his race. So you must take what happens during this sprint, and then later what's the process that occurs when he breathes in rapidly, and what's the final effect. Okay, so you must cover all these parts, all these these processes, okay, and events. Now let's start. Since it's 3 marks, so you have to explain in more detail, right? At least have 3 different points. Okay, let's look at the answer. So during the event, the sprint, the rate of oxygen used by the muscles exceed the oxygen supplied by the blood, right? So as the athlete is running very fast, he's he's increasing his cellular respiration to produce more energy for faster muscle contraction, right? So he needs a lot of oxygen for cellular respiration, more and more oxygen as he's running faster and faster. So the rate of oxygen used by the muscles in cellular respiration now exceeds the oxygen supplied by the blood through our breathing, through the person's breathing, and through the pumping of the heart. So now the muscles find that they are in a state of oxygen deficiency, lack of oxygen for the aerobic respiration, okay, according to this equation. And they undergo oxygen debt, or they incur oxygen debt. How do they incur oxygen debt? Let's find out. The muscles have to undergo another type of cellular respiration, which is fermentation or lactic acid fermentation, which does not use oxygen, okay, since the muscles are in a state of oxygen deficiency. So what is the process or the acronym for lactic acid fermentation? It's GEL. Glucose breaks down to give energy, a little bit of energy, and then plus the formation of lactic acid, which is toxic and has to be removed later on, right? So the glucose is not broken down in the presence of oxygen in this case, right? No oxygen is used. So as the person is running and undergoing the process of fermentation, together with the aerobic respiration still occurring, because the person is still breathing in oxygen, so his lactic acid levels will increase already, all right, until the end of the race. That's where he incurs the oxygen debt as the lactic acid is being produced, because lactic acid has to be removed by oxidizing it to form water and carbon dioxide after that. So that's why he breathes in rapidly. After sprinting, the sprinter needs to breathe in the oxygen rapidly. That answers the question, right? That's during the process now already, after the event. What's the process that occurs? The sprinter breathes in more oxygen rapidly in order to oxidize all the lactic acid into carbon dioxide and water. Okay, so this is the point. And the final effect is that when all the lactic acid is removed, then the oxygen debt is paid, and the breathing rate will go back to the normal rate at rest. Okay, and he has paid his oxygen debt, which is in which the oxygen that he breathes in, the extra oxygen he breathes in very quickly is to oxidize the lactic acid and remove it from the body. All right, so it's 3 marks, so any 3 points here to get the marks. Now if you're not sure, write more points, a little bit more, it's okay. All right, don't write a full essay, right? Many points, but you can write a little bit more below the lines. So here's a hot question. Diagram 7.2 shows a type of substance taken by an athlete during fitness training involving vigorous activities. So this is sodium bicarbonate. Based on diagram 7.2, justify the intake of these substances during fitness training, right? So if you have not read about the use of this substance, then you have to make a guess, all right? So sodium bicarbonate is an alkaline substance. What will it do during the fitness training? It will react with if it's taken or eaten or drunk as a solution, it will react with the lactic acid. Okay, so justify the intake of these substances, meaning that what can it do to help the person who undergoes fitness training. Okay, 3 marks. So this is a hot question. So you try to use your logical reasoning and try to give a rational explanation. Okay, try your best. Now, this is the answer. The sodium bicarbonate will increase muscle efficiency or athlete performance. Okay, because it has a high pH value, it's alkaline, it will neutralize the lactic acid or the hydrogen ion that's produced by the muscles during vigorous activity. Because lactic acid contains a hydrogen ion as an acid. It will buffer the formation of lactic acid. That means it will neutralize the lactic acid so that the condition will not become more acidic. Okay, but the best is neutralize. Just say neutralize lactic acid. That's enough. So when the lactic acid is neutralized, it's no longer able to cause the muscle fatigue. So this will prevent muscle fatigue or tightness in the athlete so that they can perform better for a longer period of time. So any 3 points here. So here's the last question, question 8. And it tests Form 4, Chapter 6 on cell division. So this is a good question. Let's go through it. Diagram 8.1 shows non-disjunction of homologous chromosomes in one of the stages during meiosis 1. So in Form 4 and Form 5, now Form 5 is in this topic of non-disjunction is in Form 5 Chapter 12 on variation. Okay, and then Form 4 Chapter 6 it does mention a bit about non-disjunction that causes the Down syndrome child. Okay, to be formed. So causes the abnormality in the gametes, and then finally Down syndrome child, okay, can be produced through that non-disjunction. So here, non-disjunction of homologous chromosomes, what is the meaning? It's a process, an abnormal process whereby homologous chromosomes fail to separate during anaphase 1 as shown here, or sister chromatids fail to separate during anaphase 2 in meiosis. Okay, in this case. So let's look at this diagram. Here you see non-disjunction occurring where the long chromosomes, this is a pair of homologous chromosomes, they move to the same pole. Okay, whereas the short chromosomes, they undergo the normal process. One of the chromosomes in the homologous pair moves to the pole on the left, the other one to the right. Okay, so the abnormality is here, non-disjunction occurs to the pair of long chromosomes, right? So in the end, after anaphase 1 is over, the cell produced on the left-hand side will have 1 short chromosome and 2 long chromosomes. And the cell on the right is also abnormal. These 2 cells are abnormal. So on the right, you have a cell with only 1 short chromosome and no long chromosomes. Okay. Now, then what happens in anaphase 2? In anaphase 2, sister chromatids will separate. So imagine the cell at metaphase 2 first. At metaphase 2, you have 1 short chromosome lined up at the equatorial plane or metaphase plate, together with 1 long chromosome and another long chromosome below the first long chromosome. So they form a single file. They are all lined up at the equatorial plane, single file. So in anaphase 2, that means after metaphase, in anaphase 2, the sister chromatids of all these chromosomes will separate, right, and move to opposite poles. So you must show, this is at anaphase 2, show the sister chromatids separating from each other for each chromosome. Okay, so it will look like this. Okay, now sister chromatids in the short chromosome, they separate from each other. And this one is in the first long chromosome, the sister chromatids separate. And then this is for the second long chromosome. Okay, now how about for this cell? This cell is quite simple. During metaphase, this chromosome will be lined up at the metaphase, at the equatorial plane like that, during metaphase 2. And then in anaphase 2, the 2 sister chromatids will separate from each other. Right? So you must show sister chromatids separate from each other for the short chromosome. So a thing to note here is that you must follow the length of the chromosomes as seen in this picture. Refer to that. Now the top one, show the short chromosome. 1 short chromosome, so the sister chromatids of the short chromosome separate. And below that is the long chromosome. The sister chromatids of the long chromosomes separate from each other. Okay, whereas on this side it must be the short chromosome, the sister chromatids of the short chromosome separate. Don't draw all the chromosomes of the same length or the sister chromatids of the same length. Okay, you'll get it wrong. So differentiate now, because long and short chromosomes, what's the difference? The difference is that the long chromosomes carry different types of genes compared to the short chromosomes. Okay, right. Complete diagram 8.1 to show the effect of non-disjunction during anaphase 2. So this one you have done it, so 1 mark for each answer. Next, diagram 8.2 shows a type of tumor that has spread to other tissues and organs in the body. Okay, so it's spread into the kidneys here, right? And the cells are cancerous. Now, it suggests a method that can be used to treat the tumor. So this is a hot question too. You have to base it on your knowledge of treatment for cancer. All right. So for every disease, even for this for cancer, you have to know the definition, the cause, the symptoms or effects, and then method of prevention and also treatment. What is the treatment? Okay, for every disease, you must know these 5 aspects. Right, so explain. So the method that's used to treat the tumor, nowadays there are 2 basic methods. All right, let's have a look. The first one is radiotherapy, and the second one is chemotherapy. Now, so you have to explain 3 marks. So explain either method. So radiotherapy, because they only ask for 1 method, so don't explain 2 methods and then just give 2 points, like F1 and E1. You won't get the complete 3 marks. Okay, so you must have 2 elaborations here. Now, radiotherapy is a method used to treat cancer. So this is the first name, the first method of treatment. Okay, then explain what it is. This method uses high-powered ionizing radiation, or just high-powered radiation. Okay, and then what is the purpose? To kill the cancer cells and reduce the size of the tumor. All right, then F1, E1, and E2 must be together. Then you get the 3 marks. Another one is chemotherapy. So if you suggest chemotherapy, then explain. What is it? It's a drug treatment that uses powerful chemicals that are injected or eaten. They're injected in the body, or the patient will eat the drugs, and then to kill the cancerous cells and stop them from dividing. All right? Same thing, to kill the cancerous cells. Next, let's go on to the next part. Diagram 8.3 shows the karyotypes of 2 individuals who have a genetic disease. All right. So this is based on Form 5, Chapter 12 on variation. Now, individual A, you can see that the abnormality is has got only 1 X chromosome, 1 sex chromosome X. Okay. So based on our knowledge, this is Turner syndrome, right? And in the second diagram, you can see 3 small chromosomes, number 21. So this is Down syndrome. Okay. So complete the table 4 to show the differences of mutations for both individuals, right? So what's the name of the disease? Okay, let's write down. The first one is Turner syndrome for individual A, and for individual B is Down syndrome. Okay, what type of cell is involved? For Turner syndrome, how is this individual produced with this type of karyotype? Okay, karyotype means a picture that shows all the chromosomes in 1 diploid cell of the person, right? So why is that there's a missing X chromosome? Is because there is a mutation or a mistake, okay, in the formation of the gametes. So the germ cells are the cells that form the gametes. So there's a mistake or a mutation in the germ cells, all right, where there is non-disjunction occurring, okay, and then it leads to the formation of the Turner syndrome child. Okay, so that will be discussed in another part, right? How does Turner syndrome child form? And for individual B, this one is studied in Form 4, Chapter 6. All right, how is the Down syndrome child formed? There's non-disjunction again in the gametes, in the formation of gametes. All right, and that leads to the Down syndrome child. So that is also the germ cell. It's not the somatic cells or body cells. So this answer is given in the, I think in the marking scheme. I'm not sure, but the right answer is germ cell. So actually, it's not a difference. That means in both cases, there was a mutation in the germ cells, non-disjunction occurred, which produced a gamete that's abnormal, with an abnormal number of chromosomes. For example, For Down syndrome, the gamete form will have 2 chromosomes 21, whether it's a sperm or ovum. Then when it undergoes fertilization with a normal gamete with 1 chromosome number 21, that's how the zygote form will have 3 chromosomes 21. Okay, so it's still germ cells that form the gametes that have the mutation. All right. So if you want to find another difference between Turner syndrome and Down syndrome, it's very easy. Just look at the number of chromosomes in each of their somatic cells. For individual A, each somatic cell has, you count the number of chromosomes they have, he or she has 45 chromosomes, correct? Because there's a missing sex chromosome here. It should be an X chromosome. So individual A has 45 chromosomes and individual B has 47 chromosomes in each diploid cell. That is a difference, right? So this is a hot question, the comparison of Turner syndrome individual with Down syndrome individual, the differences. Now we've come to the final question. The statement below shows an article from a newspaper. This is also a hot question, right? So we read the English version. The fate of Agent Orange victims. Agent Orange was a powerful chemical mixture used by US military during Vietnam War to eliminate the North Vietnamese Viet Cong troops, as well as crops that might be used to feed them. It was later revealed to cause serious health problems. Okay, this is the part. Tumors, including tumors, birth defects, rashes, psychological symptoms, cancer, among the returning US soldiers and families, as well as among the Vietnamese population. So justify the prohibition, that means the prevention on the use of chemical weapons in warfare. Okay, why is it that use of chemical weapons in warfare should be prevented? Explain why. And that is based on this passage, right? So we can see the effects of the chemical weapons. It causes the tumors, birth defects, and rashes. But to justify, you have to mention how these symptoms occur, these serious symptoms like tumors and cancer. That means the Agent Orange, start with the Agent Orange. So the event is that when they spray the Agent Orange acts as a what? Yes, it acts as a mutagen. This is from chapter 12 in the Form 5 syllabus. Okay, we study about mutagens, which are agents of mutation. That means the things that cause mutations. And then you have to define mutation. So actually this question is testing the student on mutations. That's all. Okay. So Agent Orange or chemical weapons is a mutagen, all right, which causes mutations. That means the abnormal changes in the genes or chromosomes or the genetic material. Okay, now that's the definition of mutagen. And the definition for mutation is there are abnormal changes, okay, in the sequence of the nucleotide bases in a gene. That means such as the ATCG bases, and then which are adenine, thymine, guanine, cytosine, the 4 bases. There are changes in the sequence of these bases in the gene, and then or changes in the chromosomal structure, where part of chromosome can be deleted, for example, and the number of chromosomes. Now, changes in number of chromosomes is like, for example, in the formation of child's Down syndrome or Turner syndrome. Okay, the number of chromosomes are changed. So that is the definition of mutation or the cause of mutation. And then comes to the final effect. It produces new genetic material and changes in the characteristic of an organism, and it causes tumors, cancers, and birth defects. Okay, so right, any 2 of these to get 2 marks. So actually it's not that difficult, but just that you have to know what facts are needed to answer this question. Okay, right. So that's all for this lesson. Thanks for viewing, and I hope that you have benefited from this discussion. And please do share, like, and subscribe to my channel. And if you have any comments or feedback, please write in the comment section below. Right? So we will continue with the discussion of the essay questions also later on, and to have more practice, all right, before the SPM and also for the Form 4 students before they take their final term exam. So All the best to you and stay calm and study for the exam. Bye for now.